everyone. Welcome back to U8 World Innovation Summit 2021 Spring Conference. I'm Xiongxin, a current researcher at Harvard Medical School, hosting this keynote session. As we know, revolutionary progress in science always needs persistent effort and confidence. Today, now it is my pleasure and honor to introduce a pioneer who has proven the patient, persistent, and perspiration could make an unbeatable success for world innovation. Joachim Frank. He is known for his pioneer work in Cryoelectron Microscopy, and he won the 2017 Nobel Prize in Chemistry. Such achievements come from his years of devotion to groundbreaking basics research. During 1975 to 2008, his work in the single particle algorithms for electron microscope image processing has been widely used by hundreds of labs around the world. Along with over 30 years of dedication and devotion in his field, Professor Frank's series of important pioneer work on the three-dimensional reconstruction of ribosomes finally made him win the Nobel Prize in Chemistry in 2017. Later, his work in a cryoelectron microscope makes significant contribution to the structure and function of ribosomes from bacteria and eukaryotes in the world. At the same time, he's a very open-minded and wise man. He encourages intellectual freedom and open-minded attitude towards science development. He believes science is a tool to unite people in a special way as the victory of rationalism without boundaries nationally. That's why we have Frank today to join us in discussion. Kindly remind that following this session, there is a live Q&A discussion where Joachim Frank will answer the question from the audience. So please don't forget to set your question in the Q&A box below. So let's welcome Frank to have the speak. Hi, Dr. Frank, can you hear us? Hey, Professor Frank, can you hear us?
Yes. I'm trying to share my screen.
Don't worry, take your time.
Okay, can you see it? Can you see my screen?
Yes, okay. I believe.
Hi, I'd like to talk about the way we can use cryoelectron microscopy to not just get static structures, but also get information on motion. But I will begin by talking about single particle cryo-EM. How does it work? What is it good for? I'm going to give you recent examples about viruses related to pandemics and how do we get information on molecular motion. So I should mention molecular machines in a cell. Very often we look at molecules interacting with one another, and we want to know everything about the structure, but also the way the molecules behave, how they change their structure in order to make the interactions that are necessary for the life function. And in molecular machines, many molecules act in concert in a processive way. Processive means that once a molecule engages a ligand, the ligand continues to be engaged until a certain steps of function are completed. And the concept of molecular machines has been popularized by Bruce Alberts in a very important article in 1998. Now, we wish to understand the structures of all components in the cell and the way they interact dynamically. But it is very difficult to understand them directly in the context of the cell because of the molecular packing. It's very dense packing, and it's very difficult to get with instruments of structural biology right near to them. And so we use the concept of reductionism. We study a subsystem in isolation in vitro, hoping to approximate the processes in the environment of the cell. So why don't we just use X-ray crystallography? That's been such a successful technique. Well, there's one very important reason. Crystals are not good examples of lifelike environment for molecules. They essentially act as energy traps. So molecules are then in a state from which they essentially cannot escape. This is very atypical for the way molecules work in the environment of biology. And besides, and this is a very big reason, many molecules cannot be crystallized at all. So there was actually really a need for another technique. Now, single particle cryoelectron microscopy, what it means is, I'm sorry, we image molecules by electron microscopy as single particles in solution. And the advantage is that the molecules can assume all native states. They are not sort of in a straitjacket. And in standard cryo-EM, which I want to distinguish from time-resolved cryo-EM, which I'm going to introduce later on, in a standard cryo-EM method that is now practiced all over the world, we use pipetting and blotting and plunge freezing with a cooling rate of roughly 100,000 degrees per second. And so we get a snapshot of molecules in a thermal bath at room temperature. We get sort of an instantaneous snapshot that shows all the molecules just at the instance at which they are frozen. And here's a cartoon of a transmission electron microscope, just for people who might not be so familiar with it. There's a high voltage applied here. An electron gun accelerates the electron to very high speed, to 100 kilovolts or the 200 kilovolts, 300 kilovolts. And then there comes an illumination system that focuses the beam. And then in the objective lens, the beam hits the specimen, and then the electrons get scattered. And then there is a system of magnetic lenses, which they act like glass lenses on light, but they are magnetic. And because electrons get stuck very quickly in air, there is a need for high vacuum all the way down the column. And at the very end, we have a fluorescent screen in older instruments or plate camera. And nowadays, we have an electronic camera, a digital camera. So this is the transmission electron microscope. And I just wanted to show you an EM grid. The actual diameter from here to here is 3 millimeter. So this is a much enlarged version. And what we have here for support is a copper grid. It can also be from a different metal, can be gold. And then on this, we have a so-called quantifoil. It's a carbon grid with holes in between. And optional is a thin carbon film. And then, so we have a water layer, which then turns into vitreous ice. And inside of the water layer are the other molecules. So I'm going to show you what happens with that grid. Okay, here we have the grid. We look at it with increasing magnifications. So we have molecules here, which might be dynamic, that change their structure. And now we form an image. Now the image is going to be very noisy because we have to reduce the number of electrons so that the molecules don't get damaged. What it means is there is really no clear look at any single molecule. What we need to do is average the information over many, many molecules. And so this is the typical picture that we see in the transmission electron microscope. These are projections. And then with a computer, we take if we take the individual molecules out, the way we see them, and then we essentially get a gallery of particles. And now comes the important business of computation. And this is where I made my mark several decades ago to develop programs that make sense of this information. So we have a micrograph of molecules. We do extensive data processing, and now we can get near atomic resolution reconstruction. Now, I have to mention that we don't get just a single structure, but we get several structures in one stroke. In the sample, molecules may exist in different conformation and ligand binding states, and they are very powerful programs of classification by maximum likelihood, which all can recover them separately. So we get not one, but multiple reconstructions out typically from a sample. And then I have to mention the resolution revolution. Up to 2012, the progress was rather incremental in all these methods, and the hope to get to atomic resolution was not very high. And then it was discovered that the single bottleneck was really the recording. You might say that it was a rather trivial obstacle, but that obstacle was overcome in 2012 with single electron detecting cameras. So they have completely revolutionized the field so that the technology that has been developed over the decades all of a sudden became a very hot technology that could rival X-ray crystallography in resolution. So 3 to 4 angstrom is easily achieved now for well-behaved molecules. And the record of resolution is so far 1.2 angstrom. So there is the potential for use of cryo-EM in drug development, because to get to something like 2 angstrom is really very well feasible now. And I'll just show you a couple of examples where cryo-EM has been very important. It has been important in looking at viruses, the SARS-CoV-2 spike protein. And here is an example out of many, many papers that have come out of work done by cryoelectron microscopy to look at virus structures. So this happens to be one that came out from Columbia University. So this shows the way antibodies, particular antibodies isolated for patients, bind to the spike protein, and explains the action, the neutralizing action of the antibodies. And this brings me to this slide that I've been working on just in time, namely the high resolution single particle cryo-EM, which can be traced back to 2012, has arrived just in time to help us with exploring the origins of recent pandemics: MERS, Zika, Ebola, dengue, and now SARS-CoV-2. So this is really absolutely remarkable that we have the technology that's arrived just in time. Okay, with that, I'd like to go into the actual subject of my talk, capturing motion by single particle cryo-EM. Now, this is done in two ways. It can be done directly by time-resolved cryoelectron microscopy. So this is a technique that involves a special device, a microfluidic chip. And here we capture short-lived states in the reaction of two or more components of a molecular machine. And the end product is then in thermal equilibrium. So in the end, we might still have heterogeneity, but it's the normal kind of heterogeneity. So this is the direct technique. And then there's an indirect technique, which is to make an absolutely fantastically large data collection, hundreds of thousands or millions of snapshots of molecules. When you capture so many molecules, then even states that are not very well populated become visible, and which means that we essentially then have a continuum of conformational states, and we can really then count the different kinds of conformations that we see all coexist. And when we do a deep data analysis, then we can get the free energy landscape. So this is the first experimental method that gives us a complete free energy landscape. Now, I cannot get into this today. I'm going to talk about the direct method. So I just have a slide. So in the end, from a very large data collection using these machine learning techniques, it is possible to get the energy landscape of a molecule. And from there, we can essentially infer the trajectory of the functional trajectory of a molecule. So I'm going to proceed with the other method. And I'm going to start with the timescales of molecular motion in short-lived states that was created by these authors, Fenwick et al., 2011. You can see that there is a variety of biophysical techniques that gives us information from the femtosecond to the second scale. Not mentioned is cryo-EM here. So I have created another slide with a slide modification here. So, so the important thing is that domain motions that are so important for looking at molecular machines are typically in the time scale of milliseconds. And this is exactly where time-resolved cryo-EM kicks in. So I just want to reiterate the standard cryo-EM with pipetting, blotting, plunge freezing. This is all very complicated. It takes several manual operations, even though it's computer assisted. So it takes several seconds. So it is completely hopeless to use the standard technique in order to follow a reaction in the millisecond time scale. So instead, we have now devices, and there are two approaches. One is by spraying mixing on the grid by Berryman and Unwin. This was already developed in 1994 to look at acetylcholine receptor, the way it reacts to acetylcholine. And the newer method that I have adopted is by mixing spraying on the grid, which means there is a controlled mixing and then the spraying onto the grid. Okay, here, just to recall, this was a groundbreaking paper by Berryman and Unwin, Ultra Microscopy in 1994. And here, the one of the components already exists on the grid. So it's applied to the grid in the standard cryo-EM way. So what they applied is the acetylcholine receptor in the form of helical fibers. And then this grid, this wet grid, passes a sprayer where the acetylcholine is sprayed on. So the mixing is directly done on the grid itself. So a critique of this technique is that one doesn't have a very good control over what is happening on the grid. So it is preferred to mix separately. So in the time-resolved cryo, I'm using mixing spraying. We look at short-lived states, 10 to 1,000 milliseconds. There's a microfluidic chip that was created by this author at RPI. This was on the occasion of a collaboration between RPI and the Wadsworth Center in Albany around 10 years ago. And now we are using PDMS in collaboration with the Columbia University Engineering Department. PDMS, because it is much easier to use and essentially redesign, and it's much easier than using silicon wafers. And this is sort of a cartoon that was created in a commentary. Essentially, you did this very simple idea. We have, and this is an experiment where the subunit association of the ribosome was studied just as a proof of concept. So they both are mixed in a micromixer and then reacted for a certain period of time. That's not shown here. And then with the help of a driving gas, the product is sprayed onto the grid. And then in the end, we have as usual, a data analysis after we image the molecules. We have a data analysis that looks with classification techniques at all the different structures that coexist. And here is a schematic diagram. We have a solution 1, solution 2, and they are mixed in a little compartment, and that mixing takes half a millisecond. And from then on, there is a reaction chamber which has different lengths for different chips. And then with the driving gas, the product or products are sprayed onto the grid as the grid falls into the cryogen. Now the grid is not wetted before; it's a dry grid, and it receives droplets. This is the chip right here with the two inlets. Here's the driving gas, and the sprayer is right at the end. Here are the tweezers that hold the grid. The tweezers sort of are computer controlled and fall down and are plunged down into the doer. So the application so far in the bacterial translation field, and this is all translation initiation. After mRNA is encountered, the two subunits come together and form a functioning ribosome. And I'm going to show you the experiment later on in just a couple of slides. Then translation termination. After the polypeptide is made, a stop codon is encountered, causing the polypeptide to be released. Ribosome recycling. After termination, the ribosome is split into two subunits. And in progress are studies on translation termination, initiation, recycling in eukaryotes. So here's a ribosome in a flow diagram of all the different reactions that take place. And here's particularly initiation. And okay, I'm going to show you it in a moment. But this has resulted in a paper in 2019 in Nature on the late steps in bacterial translation initiation. So the initiation is this particular place where essentially after an mRNA encounters a small subunit, an initiation complex forms with the help of initiation factors and an initiator tRNA. And so what we wanted to study is the process of subunit joining. So this small subunit that has already been preloaded now joins the 50S subunit. So here's translation initiation in bacteria. So we have these two complexes, we mix them together, and then we know from other studies that didn't involve imaging, we know that there are two complexes formed that have lifetimes of less than a second. One is the pre-70S initiation complex. You see here, it is estimated to have lifetime lower than 20 milliseconds. So it wasn't unclear whether we're going to be able to see it. But there is another one with 80 milliseconds. That's an important short-lived state, which still has initiation factor 2 present. It has a tRNA in an incorrect spot, and it waits for the release of the inorganic phosphor after GTP hydrolysis. And only after a much longer time, we get the full formation of the elongation-competent ribosome that is ready to go and do translation. So here are the different components, and I don't want to bore you with this. This is essentially what was in the slide. And here is a survey micrograph that shows where we have productive data collection in droplets. Here's a representative micrograph that shows us all the different coexisting structures. And here's the data, data collection. And so if we then look at what we are getting, we can make an inventory of all the different structures at any given time. So we have made multiple experiments with different reaction times. And if you look at all of them, we get all these different structures. One of them is the structure we start out with. Here's a reaction product that is probably on a dead end. There's a very small number of these, and that's why we have a low resolution. Here's the other beginning component. So these two went into the system. And then we have no trace of the first initiation complex, but we get the one at 80 milliseconds very strongly, at a pretty good resolution. And then we see the final product, the elongation competent ribosome. So what we see now very nicely is the way the components rearrange from here to here, and then how the next transition takes place from this intermediate state to the final state. So this has never been seen before, because any look at this involved some kind of chemical intervention. So where we don't know whether this is visible as an authentic state. Okay, so now just counting how many we have of each kind at every time point. We have the following time points: 20, 80, 200, and 600. Now you see that the concentration of the 50S goes down. The number of 50S that we count goes down. The 70S, the 70S goes up like this, and the 70S. Yeah, I'm sorry. This is the 70S that goes up. And here is the 70S initiation complex, which rises in number, and then it falls rapidly. So we have captured pretty much where it assumes a maximum at 80 milliseconds. So you can see that with normal cryo-EM, which is in the range of several seconds, there is no trace left of this intermediate state. So with our device, we were able to capture it. And so since we get particle counts so nicely, it raises the question, can we not use this also, not just for looking at structures, but also to do kinetics? Because we could do kinetics simply by counting particles at all the different time points. Well, that is a bit more difficult, because we need the reproducibility of particle picking and selection. That is a question mark. Maybe by using AI methods, we can get a more conclusive answer to this, that we get accurate counts. Now, the reproducibility of particle classification runs with different seeds. We check this. The reproducibility is beautiful. We also asserted that the insensitivity of particle classification that the particle classification is insensitive to the addition of noise. And then a further question is the accuracy of the timestamp. How wide is the distribution of reaction times that molecules experience as they flow through the channel? And there is a certain divergence. The larger the residence time, the more we get a spread of the time. So the later measurements at the hundreds of milliseconds are really not that accurate because they really draw from an entire spread of such a distribution. So here's the only short-lived intermediate captured in a bit more detail. And since I'm not talking to a ribosome crowd, you will probably not appreciate all the subtleties of this. But the point is that we get very precise information about a state that has never been seen before. And we also see, we can now see the repositioning of the tRNA in the conformation change of initial factor 2. So in conclusion, the experiment revealed the kinetics of the reaction. The pre-70S initiation complete could not be captured since one of the initiation factors leaves before 20 milliseconds. And so the apparatus was unable to capture this. The 70S, the 70S IC is seen for the first time. It's still in the GDP PI state. And the release of IF2 is accompanied by small intersubunit rotation and initiated tRNA repositioning in going from the intermediate state to the elongation-competent ribosome. So in conclusion, overall, cryoelectron microscopy is still evolving. It's not just in the direction of higher resolution. Cryo-EM is uniquely suited for capturing native states of a molecule. And short-lived states of any molecular machine can be visualized at a spatial resolution comparable to standard cryo-EM. Time-resolved cryo-EM can be used to determine kinetics after these additional investigations that I just mentioned, just by counting the numbers of particles in subpopulations as a function of time. And what I didn't mention is that there are efforts now going on to redesign the experiment and redesign the apparatus to be able to go down to 1 millisecond. And that would be very exciting because we would be able to follow the switching and gating of ion channels and receptors. Thank you very much.
Well, thank you. Thank you, Professor Frank. Thank you for a great speech about the cryoelectron microscope with us today. We've known that cryoelectron microscope could be used for structural analysis of dynamic biological macromolecules. Right now, I'd like to move this part for the audience open questions. There is an interesting question from the Q&A box. He's asking, Rose is asking, in this methods, how to distinguish the signal of amorphous part in the molecules from the amorphous background.
How do I distinguish the signal from the amorphous background? Well, I pointed out before that the molecule itself, as it appears in the electron micrograph, is very noisy. So there's absolutely a need for averaging. So this is the main reason why we collect such a large number of particles. And so through the use of averaging, the noise is eliminated and the background is eliminated. So the background, if with the background, this person means the background on the grid, it is also regarded as noise by the algorithm because it's ever changing from one particle to the next.
Mm-hmm. Okay, if rather you have following questions, you can ask in the Q&A box below. I'm gonna move on for the next one. So he asks, would PDMS be moved from the specimen in the end? Would be generate similar contrast with biosamples in the image, and how to separate them from your experience?
I'm sorry, I didn't get that question now.
Well, probably you can open the Q&A box. So the audience is asking, would the PDMS be removed from the specimen in the end? Would be generate similar contrast with biosamples in the image, and how to separate them?
No, this is this is a misunderstanding. PDMS is used as the material to make the actual channel, the microfluidic channel. This is PDMS. All the walls are made by PDMS. They are not inside of the solution. Mm-hmm. So there's no separation necessary. It has nothing to do with it. It only constitutes the walls of the device.
Mm-hmm. Oh, back to the question that the audience Rose is asking about the amorphous background. She's asking about an amorphous background of ice.
Yes. If you let's say we have different realization of the same molecule, and they're all facing the same way on the grid. Okay, then each of them has a different ice background. Okay, so if you characterize the ice background as some kind of a noise function, that noise function is going to be different everywhere. So it averages out together with all the other components of noise.
Great. Thank you, Professor Frank. You told us that cryoelectron microscopy revealed the structure of proteins by pumping a flush frozen solution with a beam of electrons, right? And then combining 2D image of individual molecules into 3D pictures. But do you think the cost of such a high energy microscope will limit such methods adoption from your perspective? What do you think?
Well, it's the I mentioned in the beginning that the electrons interact very strongly with the molecule, so they damage it, and this is why we keep the number of electrons that the molecule sees very much contained, very low. So we keep the damage to the individual molecules very low, and this is why we see the molecules like pepper and salt. Okay, we don't see it very clearly, and this is why we need such a large number to average over.
Thank you, Frank. So alongside with your scientific great pursuits, I have known that you have maintained lively devotion to the arts of creative writing. I even write some novels, right? Can I ask, do you find the science and your art kind of relates in any way?
No, it's all one piece of the same, really. But I find that the pursuit of science is so dry that I need to do something else in my life. So I discovered that that with art and literature, I can find a balance in my life. But in some way, I mean, there are very interesting relationships between the two pursuits. So one can inspire the other.
Yes, I totally agree. So do you think when was your scientific interest sparked in the beginning? Because I'm also like working on the science field, even though it's in healthcare, more about neuroscience. Always very curious, like a pioneer like you, when was your scientific first sparkling like interest you to work on this?
Yeah, it was really, it came up before I knew what science was. I was 8 years old. So I did all kinds of experiments under in a space under the veranda of my parents' house. And so I collected bottles and filled them with all kinds of liquids and experimented with them. So it was really a complete native interest that I always had in nature and in things around me. And that sort of more crystallized into an interest in science when I later got into middle school and high school. And then I have to mention, I have to give credit to a very inspiring teacher. And I had a very inspiring physics teacher. And so this is one of the reasons why I got so excited in the whole field.
Oh, yeah, that's impressive. I believe a lot of scientists who work on this field always have a very good mentor in their beginning, right? And I'm very impressed that actually during the 1975 to 2008, there is over 20 years, you've been very dedicated on your work in the single particle algorithm for electron microscope. But at that time, you haven't had the chance or honored, like publicly honored your work. And in 2017, you suddenly get the Nobel Prize in chemistry. So I was just wondering, like, how well do you know your fellow you're gonna win the chemistry Nobel laureates in 2017? Do you have any idea before that time?
Well, I think I had an idea that there were nominations floating around, and it you know, among all prizes, it could only be chemistry. I mean, because physics is really dedicated to completely different things like black holes and whatever. And medicine is also very, very much more in the clinical realm. But my the fellow people, Dubuchet and Henderson, they are physicists, so they really didn't come from chemistry, and for me it was really bizarre. I was physicist, and then I worked in biology, and all of a sudden I get a prize in chemistry. I always hated chemistry in high school. I couldn't relate to it, and you're being regarded as a chemist when you invent something that actually reveals structure, you know. So that's why I became a chemist in the eyes of the people who award.
Yeah, I'm very impressed by your work. I think you are a great combination from the physics and chemistry, such a combining all. Like always, I saw a lot of interview from you. Like I think you really encourage the fellows about intellectual freedom, which means there's no boundary nationality or from even the subject. So you really encourage us to think about combining different subjects and ideas together, make new innovations. And one last question. I'm wondering, so how was life changed since you received the prize? Is there any collaboration internationally becomes more frequent?
No, not really. Life changed in a way that I was able to meet people that I would not have met otherwise, and that's more in the realm of completely different fields. I mean, it was amazing to get the acquaintance of Ishiguro Kazuo Ishiguro, the literary Nobel Prize, and so many other people that I would not even have had an opportunity to speak to, all of a sudden are open to conversations, and so that's a very big deal. And in terms of science, I felt more encouraged to tackle something that was more adventurous, that involved more risks than before.
Mm-hmm. Oh, there is a new question from the Q&A box. They'd like to, Professor Frank, you answer. So from the audience, Mark, he's asking, does your technology have any limitations of molecules, such as molecular weight or etc.?
Yeah, that's very important. The molecules need to be big enough. And it used to be that there should be at least something like 150 kilodaltons. But more recently, because of the higher sensitivity of detectors and all kinds of algorithmic improvements, that limit has gone down to maybe 50 kilodaltons. But if you get lower than that, then it becomes very, very hard. And there are methods. If you want to get a structure of a very small molecule, then you can consider attaching it to a larger molecule that you already know. So it will be reconstructed along with a larger molecule. So this is something that is definitely feasible.
Mm-hmm. So, Professor Frank, do you think what's the next step for the supervision microscope? This is asked by another audience, Drew Yu. What do you think the next step for the super resolution microscope, and what is the current biggest?
Oh,
yeah, super resolution microscope. And what's the current biggest contender from achieving it? Like biggest obstacles to achieving it?
To achieve what exactly? We're already at 1.2 angstroms. What exactly do we want?
Probably she's trying to ask, so what's the next step for the super?
Well, it's really, I think it's really not going higher in resolution, but rather doing something with the technology, as I showed, the two different things like time resolved technique and then this machine learning technique, which is able to recover energy landscapes. So this is where the innovation should be, and not to achieve even higher resolution. Higher resolution than 2 angstroms is really to the benefit of material science. And they already have microscopes that can do a very, very good job. And so, but as far as biology is concerned, I think we have to be concerned with making this technology more widely available, so that it's not just in these very very concentrated centers, but also in a normal PI's place. So I think there are efforts making machines that are with lower voltage, and they are specialized in cryo, and I hope this is where the future leads.
Yes, I understand. Like current stage, maybe we want more to improve the applications of the existing microscope to make it progress, right? So, for example, like you definitely continue research, there are a lot of new kind of young researchers coming into your field. Do you think, what did you learn from your meetings, like with young people, young researchers recent days?
I sort of, I missed the question. I was, yeah.
What do you learn, for example, what do you learn from your meetings with young people or young research nowadays in your industry?
Well, it's really, I mean, they sort of approach the whole thing with an open mind, and but they are. I mean, what I always learn is that there is not much of an appreciation for the past, for what has gone into it in order to get this to this stage. I mean, the younger generation is now a generation that is used to apps. You know, everything can be solved by an app. Okay, so they, you know, I mean, they might even, without knowing anything about the whole field, they might ask whether there's an electron microscope app in a cell phone. Okay, so so it's really the appreciation of science history and technology development is really something that that is very important for people to see what their own potential is, and how they can see their own career and progress later on. And there's a lot of naivety.
Okay, yes, I totally agree. I think nowadays, like young people's always like to combine, like as you mentioned, apps or kind of current internet, kind of small inventions with the existing technologies.
Yeah.
I think it's a very good merge, right? So thank you very much, Professor, for your great sharing in these sessions. We really appreciate your time. And now we probably going to move to our next sessions. So thank you for your time. In a couple of minutes, we'll have the next session about pediatrics from the Harvard Medical School. So let's stay tuned and hope to see you again.
Okay. Thank you very much. It was a great pleasure.
With pleasure. Thank you.